Okay, we're going to wrap up Chapter 9 and would like to begin by just having you note that there are two different kinds of skeletal muscle fibers, the so-called slow twitch type 1 and fast twitch or type 2. I'm going to let you just read over this slide and make note of the differences between slow and fast twitch. When we look at all of our skeletal muscle, that we all have a combination of both the so-called slow twitch or red fibers and the fast twitch or white fibers. We talked earlier about how one can train to increase one's endurance, and how training involved increasing the number of mitochondria, increased vascularity, increased glycolytic enzymes, and that kind of thing. But we also said at the very end of that particular slide that one's genetics also has a role to play in how efficient or effective one is as an athlete. And so if you note here, for example, for the slow twitch fibers, that there is a predominance of many mitochondria and a good blood supply, so these fibers can contract for longer periods of time without fatiguing, which is why, for example, somebody that has a higher proportion, let's say, of slow twitch versus fast twitch, that individual might be more effective as a marathon runner, as opposed to, say, somebody who has a higher proportion of fast twitch than slow twitch might be a better sprinter than they would be a marathoner. So this gets back to the idea that our ratio of fast to slow is genetically inherited and predisposes one to maybe be a better marathoner versus a better sprinter. So we're pretty much done talking about skeletal muscle. Let's talk next about smooth. This is the second, remember, of the three types, cardiac being the last. Smooth muscle fibers look a little different than skeletal muscle fibers. They're tapered at the ends. There's only one nucleus per cell or per fiber. Remember, skeletal muscle has multiple nuclei in each fiber. And so I'll again let you look over these different bullets to kind of see how smooth muscle cells or fibers differ from skeletal muscle. And you can see that there are some pretty significant differences. And these have impacts on the physiology of smooth muscle contraction versus, say, skeletal muscle. There are two major categories or types of smooth muscle. The so-called multi-unit smooth muscle shown here, visceral smooth muscle. And again, I'll let you look over the differences, pretty self-explanatory. When we compare smooth muscle contraction with skeletal muscle contraction, we note that both have both actin and myosin, myofilaments, both use calcium and ATP, and both rely on the release of calcium to trigger the contraction process. Important differences between skeletal and smooth muscle, not the least of which is the fact that in smooth muscle we don't have troponin. If you remember, this was one of those proteins, one of those three protein subunits on the surface of the actin. You don't have that here with smooth muscle. You have another protein called calmodulin that accepts the calcium. Calcium binds here to the calmodulin protein of the actin. The other major difference you notice is the fact that unlike skeletal muscle, which relies on calcium that's stored in their sarcoplasmic reticula, in smooth muscle, the source of calcium comes from the outside. This extracellular fluid is what bathes the skeletal muscle tissue. And for that reason, the SR is not nearly well as developed in smooth muscle fibers as it is in skeletal muscle. We also described one neurotransmitter back in skeletal muscle. That was acetylcholine. Here you see, of course, there are two types in smooth muscle, ACH and NE, or norepinephrine. And while you'll be hearing more about this coming up when you take ANP2 in the nervous system, these two neurotransmitters in smooth muscle can sometimes be excitatory. In other words, can promote subsequent contraction, but in other situations they can be inhibitory. In other words, they can prevent smooth muscle from contracting at times. And I'm not going to get into the nitty-gritty of that right now. We won't discuss that at this point, but you'll be hearing more about it again in a Hormones play an important role in impacting certain types of smooth muscle, and here we see an example of that, whereby in a pregnant female who's about to give birth, as we all know, when the moment of birth comes, the magnitude and the frequency of contraction in the smooth muscle lining the uterus increases. And if someone is not progressing along well in labor, sometimes they will prescribe an artificial oxytocin called Pitocin. Usually it's given in an IV, and that can really ramp up both the intensity of contractions and the frequency of contractions. And some of you, I'm sure, who've had kids via normal vaginal birth can speak personally to the effects of Pitocin, but it really promotes some significant contraction to help that baby get born. Another really interesting property of visceral smooth muscle is the fact that when it's stretched, that can trigger contraction. And this makes perfect sense if you think about, where you tend to find visceral smooth muscle, which is in the lining of the gastrointestinal tract, the stomach, for example, and the small intestine. It's the stretching of those smooth muscle tissues that can trigger increased digestion. So when you eat lunch and your stomach fills with food, the wall of the stomach, of course, is distended or stretched, and that alone triggers the mixing and churning that occurs in the stomach. It's kind of interesting. Smooth muscle is also much slower to contract and relax than skeletal muscle is. It tends to resist being fatigued. Skeletal muscle fatigues much faster than smooth muscle does, and it has to do again with the anatomic differences between the two types. And so now we kind of wrap up the muscular system by talking a little bit about cardiac muscle, which you know is only found in the heart. And this photograph that you see on the slide, you've also viewed in lab a couple weeks ago when we did tissues, if you recall. And we talked about these structures called intercalated discs, which is where two cardiac muscle fibers come together, where they sort of meet, would be this intercalated disc structure. It's like a little line that sort of runs transversely. The fibers are running, left and right here, and then we see occasional intercalated discs sprinkled throughout the photograph. And it's across these intercalated discs that ions can flow during the time in which the muscle is being stimulated. And this is really important because if you think about, for example, the left ventricle of the heart, and we'll be getting more into the heart in a couple more weeks, but you don't want parts of your left ventricle to contract while other fibers don't. You want that entire mass of muscle tissue to contract together. And so as a result of having these intercalated discs and the movement of ions across those very, very rapidly, this entire mass of cardiac muscle contracts in what's referred to as a syncytium. There's all or none response. These are all those cardiac muscle fibers contract or none of them do. You don't want 80% contracting and having 20% not contract. You want 100% to contract when they need to. And again, that occurs for the most part as a result of ions that are moving across those intercalated discs from one fiber to another. We'll talk about the fact that cardiac muscle is self-excitatory. That's kind of interesting. It can kind of contract all by itself, although we will describe how nervous innervation can also play a role in promoting contraction of muscle, cardiac muscle. And the rhythmic aspect, if you think back to that video that I showed you in lab that first day, remember there were those two cardiac muscle cells in the Petri plate that were sort of contracting independently, but once they touched, do you remember what happened? They started to contract in synchrony, in rhythm. They were rhythmic. They were contracting together. And then lastly, this very interesting property that cardiac muscle cells exhibit, and that is the fact that you cannot induce tetany in cardiac muscle. We know that if we stimulate skeletal muscle with increased frequency, that we can get this summation and tetanic response. Go back to that video where I talked about that. In cardiac muscle, you don't get this prolonged, sustained tetany. So that's kind of interesting, and it is due primarily to the significant anatomical and physiologic differences between cardiac muscle and skeletal or even smooth muscle. This is just an interesting electron micrograph that gives you a better view of the intercalated disc between two adjacent cardiac muscle fibers. Our last slide is a summary of the three types of muscle tissue: skeletal, smooth, and cardiac. This is just a really nice kind of summary table to look at if you're interested in seeing what the major functional differences are. This, of course, is listed here, as are some of the cellular characteristics that make skeletal, say, unique from smooth, which is different from cardiac. And finally, there are also some characteristics related to the contraction of these three different types of muscle tissue. So just a nice summary table to check out that compares all three. And this voiceover pretty much finishes up our discussion on Chapter 9, Muscular System.